In this video, we will discuss about how these microbial metabolites can regulate T cell function, not just through the cell surface interactions, but also the microbial metabolites can interact with the intercellular uh, receptors in the T cells. So we will start with this cell surface receptor signalings. So most of the T cells has these GPR43 reaction receptors that can interact with a small chain fatty acid, and these interactions might triggers this differentiation of the T cells towards these regulatory T cells. And on the other hand, it can also detect this inosine, which comes from the bacteria through these A2A receptors, and this helps the differentiation of T cells to Th1. This is basically to counter any pathogens that have been detected by the T cells. And some of the receptors that are also known to trigger the change in the naive CD4 plus T cells are G2A and S1PR4. They can interact with this N-acyl amides from the bacteria. However, how they regulates the T cells is not yet been completely known. And in terms of the intercellular signalings or intercellular receptor signalings and transcription regulations, some of the metabolites can also interact with the intercellular receptors. For example, this indole 3 lactic acid can interact with this AHR to inhibit this THPOK, and this might cause the upregulations of the CD4 plus T cells towards and also the CD8 plus alpha alpha, and it will also lead to the secretion of cytokines such as IE. So I'm not able to recall what this IEL means. You can refer to the paper to understand these things in better. And also this ROR YT, it can interact with this secondary bilic acid, bile acids such as and that might leads to the suppression of this Th17 cells, or it will suppress the differentiation of the T cells towards this Th17. And this VDR also interact with the secondary bile acids such as LCA, and will also suppress the Th1 differentiation of T cells towards Th1. And in terms of this epigenetic abilities or things that will regulate the gene expressions such as secondary bile such as isoallole LCA, it will cause the FOXP3 regulate the FOXP3 expressions that will cause the upregulations of these regulatory T cells. And the small chain fatty acid prevents this HDAC or interacts with this HDAC to form the high CD8 plus cytotoxic cells. So when they interact with these CD8 plus cells, it will make the cells more cytotoxic in nature. That is SCFA, small chain fatty acid, interact with this HDAC. So this in this case SCAF inhibits, or this interactions with this HDAC gets inhibited, or its interactions will cause, so it's not inhibited. This SCFA's interactions with HDAC cause this more of this CD8 plus T cells to form cytotoxic in nature. And in terms of the metabolic regulations, the mTOR plays a main role. That is, for CD8 CD4 plus T cells, they mTOR regulates differently, and for CD8 plus T cells they regulate differently. And for this bacterial derived serotonin, when it's interact the serotonin, mTOR will get activated, and it will prevent the Th1 and Th17 differentiations from T cells, and at the same time it will promote this regulatory T cells differentiations. And spermidine, it will also promote the mTOR towards this regulating T cells differentiations. And this SCFA, this small chain fatty acid, when it interacts with the mTOR, it will also cause this Th1 T regulatory cell Th17 differentiation. At the same time, this Th17, when cells are formed, it will cause upregulations of the cytokines such as interleukin 10 expressions from TH17 cells. And when it comes to the CD8 plus cells, the SCFAs will upregulate or regulate through this acetyl CoA to form CD8 plus cytotoxic T cells. And this is the kind of effector functions of the differentiated T cells. So these are some of the relationship or how the microbial metabolites regulates T cell functions. If you need to understand more about their mechanisms or relationship in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.